Together, we are helping to improve the quality of life for patients in our community through advancing knowledge, treatment, diagnosis, and improving the care for future generations. And I think that's truly a worthwhile cause and a good way to be spending our time.
So on behalf of Shoppers Drug Mart, the Shoppers Drug Mart stores in Kingston, we're thrilled to sponsor another year of the Women's Giving Circle. I've been attending these meetings as a representative of Shoppers, and I go back to my colleagues and I tell them how great you guys are and how we should continue to support you. But recently I put my money where my mouth is, and I joined the Women's Giving Circle. It's been great to be a part of this group, so if any of you guys are thinking about joining, I certainly encourage you to do so.
This fall lunch, the Women's Giving Circle has 2 lunches, one in the fall and one in the spring. And in the fall lunch, we enjoy lunch together, but our major task is to vote on what area of research you would like to see the Women's Giving Circle contribute to in the upcoming year. And last year, as you heard from Marg, it was on aging. And this year you have 3 options, and we're going to choose which option that you would like today. And that's what this green envelope is for. And we got together in advance of today's lunch to determine what 3 options there would be. And then once you vote, basically it's the majority of votes will identify the topic that the funds of the Women's Giving Circle will go to support in the upcoming year. So your 3 options are chronic pain, parental and maternal health, prenatal and maternal health, and mental health. So let me just talk a bit about these 3 areas. First of all, chronic pain and pain management. It's very much a mystery. This is where the importance of research comes in. For pain management, there are many different types of research opportunities, such as looking at ways that certain types of pain impact patients, examining different ways of treating different kinds of pain, and examining mechanisms such as finding the optimum combinations that medications must be placed in order to work in various situations. So that's one area of needed research and one area that you can choose as the option for the research that will be funded going forward this year. The next one, and also very important, is prenatal and maternal health. And many of the studies currently being carried out in maternal medicine are related to the overall cardiovascular health of women. And researchers are finding that the time of pregnancy in a woman's life can provide a window into the longer-term health of her heart and also of the infants. So that's another area of focus or potential focus for the funding of the Women's Giving Circle for this coming year. And finally, another very good alternative is mental health. Over the past decade, a number of mood disorder studies have been carried out in the areas of depression, bipolar disorder, and anxiety. And research studies different kinds of treatment methods, medications, stigma, and much more, all with a focus on changing practices.
I'm going to talk about targeting mitochondria to treat lung cancer. And so this is me talking to you. So tell me again about mitochondrial problems in cancer, Stephen. And I say, do you want the simple but misleading explanation or the one you won't understand? And you say either one is good, I wasn't planning on listening. So, that may be the high point of the talk. So I'm assuming most of you are probably not thinking about cells on a daily basis, but the cell is really a microcosm of your whole body. What's going on in the cell is amazing, and in a moment I'm going to show you a movie of mitochondria wandering around your cells as you sit here with me right now. But I'll just, for the purposes of this talk, I'm going to just tell you that you may know that the nucleus is where your DNA is, and that's controlling the genetic profile of the cell. What I spend a lot of time studying are these little organelles called mitochondria. And traditionally, for many years, these have been thought of as power plants of the cell. So they make a chemical called ATP, which gives you energy so you can get up and move. And they do that, but there's a revolution right now, and we recognize that mitochondria are also controlling how the cells divide, and they offer an attractive candidate for diseases like cancer, where cells are dividing too rapidly. So this is the traditional view. Job one for the mitochondria is to make this energy molecule called ATP. And that's not really the chemical structure of it, but in any case, good enough. So what has been recognized by many people is that the mitochondria not only make energy, but they also kill cells. And there's this very neat And now this joke goes better for faculty member, but if you've ever been in there's a process called apoptosis, which is programmed cell death. And you may not have ever heard of apoptosis, although you've had it happen in your body before. It's what actually, when you were a fetus, your hand looked like that, and apoptosis killed the cells between your digits and gave you individual digits rather than a flipper. So you've all experienced apoptosis, but it's a way of quietly killing the cells, and I tell cardiologists and faculty members that it's sort of like a faculty meeting where there are some faculty that need to be taken out and quietly killed for the good of the group. They want it to happen quietly without a lot of noise, and that's what apoptosis is. And it turns out the mitochondria recognize a bad cell, and they will kill that cell. So this is teleologic thinking, but if you're a cancer cell, the last thing you want is to have a mitochondria kill you, right? You want to survive. Cancer cells are immortal. If you've ever read that wonderful book, The Immortal Life of Henrietta Lacks, you know that her cells, this woman who died in Baltimore more than 50 years ago, her cells are growing today. So cancer cells are immortal. They don't want mitochondria. They figured out how to shut these things off, and we're determined to turn them back on. So if you want another more CSI look at mitochondria, high-level talk. So well detected, and they're trying to figure out how this cell got killed over there on the far side. Looks to me like an open, shut case, Jim. Apoptosis. So this is the cell killing itself. And oddly enough, to make the patient live, we need to kill the bad cells. And most research has targeted the nucleus, trying to stop cell division. We're targeting the big bad mitochondria we've seen here destroying Manhattan, and we're trying to do that. And so, Your Honor, my client is an eminent scientist, and he pleads not guilty on the grounds that his mitochondria made him do it. So what does the word mitochondria even mean? This is a scientist, Carl Bendit, and he observed these mitochondria. Sometimes were little balls, and sometimes they were long threads, and so we called them mitochondria, from the Greek mitos, thread, and chondrion, granule. One of my best trainees, who I'll mention later, is a guy named Evangelos Mikalakis, who's at the University of Alberta, and he's great. We've debated many times about which language is superior, the Greek language or the English language, and of course I'm maintaining that English is because we've stolen all the Greek words that we wanted, and we have everyone else's words as well. But he maintains that the origin of everything is Greek. Since this is the woman's circle, I thought I would show this woman. This is Lynn Margulis. I don't know if any of you have heard of her, but she had this crazy idea that mitochondria were actually bacteria that came to inhabit our cells, which is proving to be largely correct. So at one time, and I'm going to show you a picture of them moving in a minute, and you may believe they look like bacteria. They actually move around, they divide, and this is all happening in all your cells while you sit here and eat lunch. But Lynn had this idea. She just died last year. She submitted her idea to in this paper entitled "The Origin of Mitosing Eukaryotic Cells," a pithy title. And it was rejected about 15 times before it was finally accepted. And so when it got accepted, she had the happiness of believing she had this cool idea and it was all hers, only to find out there was some scientist in 1905 in Russia who already had this idea, and he too was also dismissed as a bit of a nut until the idea was finally validated. That's another recurring theme in science, is you're an idiot until all of a sudden someone replicates your work and then you're a genius. So this is a picture of what's going on in your cells. This was taken with a microscope that cost $1 million. So one of the other things about having scientists like me in the community is we're expensive. And I came to Queen's in part because of the opportunity to compete for a Canada Research Chair, to build a laboratory, and I see patients, and I teach, and I administer the Department of Medicine. But these tools to run a lab, the budget for my lab is like about $1 million a year for personnel. The equipment costs about $3 million. So, and I don't compete with people in Kingston. I don't compete with people in southeastern Ontario or Canada. I'm in competition, like most scientists, for money and publication globally. And so that's the challenge of affording people like me. In fact, Richard Resnick, when he recruited me, said, "I want you to know you're not in Chicago." I said, "No, I'm not in Chicago." But it is true that I intend to play at the same level on a research basis. So that gives a big challenge for the hospital foundation. So at the beginning of this movie, what we see is we've taken the jellyfish. You know, jellyfish are fluorescent, and they have a protein called green fluorescent protein. And that gene, you cut it out just like it's a piece of material, stick it into the cassette of your vector, and we've infected mitochondria in a cell with green fluorescent protein from jellyfish and red fluorescent protein from the jellyfish. And then at the very beginning of this movie, which will start in a second again, we turn on one mitochondrion with a laser beam. And I'll say right now, there's one mitochondrion. Now it divides, and now it joins with the red mitochondria and makes a yellow mitochondria. So it's red and green make yellow. So pretty simple, but pretty cool movie. And it turns out that the punchline for this and how it relates to cancer is for a cell to divide, the mitochondria have to divide. And we figured out how the mitochondria divide, and we stopped them from dividing, and so the cells stop dividing. So I just wanted to go back to something clinical now. So this is a patient with lung cancer, and unfortunately, if any of you have had a person that you know with lung cancer, often they're fine, and then all of a sudden they're dying. And in many cases, it presents just like this. This is a CAT scan, and you see the person has a huge tumor here. And the nature of these tumors is they just show up late. People don't know they have them until they're big, and often people with lung cancer are sort of stigmatized. People, are sympathetic to people with breast cancer, but they somehow feel people with lung cancer got it themselves by smoking, and there's a lot of judgment that goes on. But if you look at this, and this is an American slide, but it applies in Canada as well. If you look at the causes of death in women, coronary heart disease is number one, whether you're white or black. Stroke is number two, but the third cause is not breast cancer, it's lung cancer. So it's not a matter of competing to say which disease is worse, but I think women tend to forget about lung cancer, and they are at risk of it, and a lot of times it's not related to smoking, and even if it is, that doesn't mean you want to die. Now, how do we diagnose cancer? And this is the tie back to mitochondria. So if you ever have cancer, you may be referred by your doctor for what's called a PET scan. You can see here on a CAT scan, which is this one, the black and white one, there's a little arrow that points to something you can barely see at the bottom of the screen. That's the tumor. Whereas on the picture that looks very similar beside it, you can see this really bright spot, which is obviously easy to identify. What that is a positron emission tomogram. It's a PET scan. We have one of those types of machines, and what happens is that in fact, I've usually had one for rats, but in any case, the cancer eats a lot of sugar. And it eats a lot of sugar because the mitochondria have been turned off, and the cancer cell is living off a diet of sugar. And this guy, named Otto Warburg, is a Nobel laureate who recognized this back in the 1920s. Otto Warburg was a German scientist who observed that cancer cells, even when there's lots of oxygen around, eat a ton of sugar. He didn't know why, but he observed that they just ate a lot of sugar. And it is that flux of sugar into the cell that we track with radioactive sugar that shows up on these PET scans. And that's why when you have cancer, usually your doctor will refer you for a PET scan so they can see where the cancer is other than in the obvious places. So Otto Warburg knew this. So it turns out that lung cancer, it's got a nice picture, has a sweet tooth. And it uses glycolysis, which is the name for the biochemical reaction of making your energy by using sugar. And so we thought that if we turn back on normal metabolism, we might be able to kill these cancer cells. So this is the question we set out to ask. This is called a hypothesis. Can we turn mitochondria back on and kill cancer cells? This is the kind of roadmap that a researcher will make, and I'm not going to take you through this other than to say the little box up here, that's glycolysis. That's glucose coming into cells. That's what you see on a PET scan, and it just stops there. Normally what's supposed to happen is that glucose not only goes into the cell, but it goes all the way into the mitochondria, which is this green thing, and it gets metabolized. And although it may be boring and it may be tedious, this is just the way it is. You have to figure out what exactly is going on. And of the many things that are going on, one thing is this enzyme right here called PDK, pyruvate dehydrogenase kinase, is turned on in cancer, and that's shutting off the mitochondria. And so we found a molecule that would turn it back on and turn on the mitochondria. So the concept is not that hard. Something is turned off in cancer, we're turning it back on to reactivate the mitochondria and kill it. This is the molecule, and if you watch, this has been led by one of my former trainees, who's now a full professor out at the University of Alberta, Evangelos Mikalakis, and has been on everything from Glenn Beck to in science to all over the news. People are very interested. In fact, if you type in Evangelos Mikalakis or Dichloroacetate, you'll see YouTube movies with hundreds of thousands of hits related to this, because this is a non-patented molecule that is cheap and readily available, and therefore in which industry has no interest, but it is very effective at treating cancer. At least it appears to be. Here's an example of how we do it in the research lab. So if you like rats, I apologize, but we use small animals like mice and rats. We treat them humanely, but there is no substitute in 2013 for small animal research, short of doing it in patients, which we also do, but you can only do certain things in patients. So this is a nude rat. It has no immune system, so we inject human cancer cells into this rat, and we watch it over time. This is an MRI in a rat, and you can see the tumor grows to be quite big. There it is under the skin. And here's a rat that was treated exactly the same. Cancer was put into the animals, the cancer cells start growing, but that rat drank this drug, Dichloroacetate, and you can see the tumor is much smaller. And at the end of the study, we cut it out, and you can see there's the DCA drinking rat, Dichloroacetate drinker, and there's the control. So that's a clinical trial in a rat, and that's usually a first step towards a clinical trial in a person. So my friend Evangelos Mikalakis did that clinical trial. Because although people die of lung cancer, there are more treatments for lung cancer, and so often you'll be told it's not ethical to give a drug to patients with lung cancer, even though we know their natural history is not very good. So we took a tumor which has got such a bad prognosis that there wasn't a lot of argument about treating these patients. This is called Glioblastoma multiforme. It's a big word, but basically it's a kind of brain tumor. The average survival is 8 months. There is only one approved drug, and it's not particularly good. And this is an MRI of a patient's brain, and you can see this white thing is the tumor. And the PET scan, the glucose scan that shows these cells eating glucose is shown right here. And you see that it's taking up a lot of glucose. This is after that patient who's now already outlived his life expectancy. You can see the tumor is not gone, but it's smaller in the lower panel. If you compare the top versus here, it's a little bit smaller, and there's a little less glucose uptake. So that's the end of the first part. That's one discovery, and we patented that, and we brought it to Queens and Partech, which is a patent group, or an IT commercialization group here at Queens, and we've put that patent with them, and we hope that they can find someone that will do something with it, because we have day jobs. So anyway, that's one thing that Evangelos and I have done. This is a picture of a cancer cell where we've color-coded. The nucleus is a black hole. We color-coded all the mitochondria that are individual into a different color, and we compared it to the normal type of cell that should be in the lung, which is this one. And you can see there's a lot more little colorful particles in the cancer cell than there are in the normal cell. And there's a picture down below again of a single cancer cell with fragmented mitochondria versus the normal mitochondria. So we asked the question, why are they fragmented? Well, when I went to medical school at Queens back I graduated in '81, we were taught mitochondria just made ATP. It was like a butter knife that served one function. But we now know that mitochondria are more like this. They have a lot of functionalities, one of which is to kill the cells, and the one that we just discovered in the second part of this talk is that they control cell division. And so there's a picture of a cell divide, and they literally have to split into two. So I guess in retrospect, you don't have to be that smart to figure if the cell divides into all the little bits that I showed you in the first cartoon, all the organelles have to divide into as well. The trick was deciding what were the proteins, what mechanisms were they. So I'm now going to, in the last 5 minutes of this talk, turn you into an expert protein chemist. So we'll see if this works. So there are two proteins that I'm interested in. One is called mitofusin 2, and one is called DRIP1. Mitofusin 2 is like Velcro. So Velcro is that wonderful stuff. There's an electron micrograph of Velcro. If you've ever used Velcro and wondered why it sticks, it's a hook and a loop. And the two things come together like a cockle burr, and they stick together. So we use those at age 6 and age over 60 to get our shoes on. So that's how mitofusin 2 works, and actually the little protein actually is like that. It has hooks on both ends, and the two of them come together, they lock, and the mitochondria dock, and then they join together and they fuse, like I showed you in the movie. DRIP is like making sausage. So DRIP1 is a molecule that when the cell gets sick, it moves from the cytosol to the mitochondria, and it makes a necklace, just like that picture down below, and it's like making sausage. That necklace crimps off the mitochondria, just like you're crimping off a sausage, and it breaks into 2 sausages instead of 1. So it turns out that in cancer, there's a lot of DRIP and not enough mitofusin 2. So that's a simple imbalance between fusion, which is joining together. There's not enough of that, because there's not enough mitofusin 2, and there's too much fission. So we wondered if we just play, so you don't have to be a genius. I mean, I could probably take any one of you and have you work in my lab, and you would know the next experiment is, well, why don't we give back mitofusin 2 and see if it's therapeutic, or why don't we block DRIP and see if it's therapeutic? And so that's exactly what we did. And obviously the fact that I'm standing here tells you that it was effective. So this is a normal cell. And as soon as we turn on the mitochondria, look how that green signal spreads immediately throughout the mitochondrial network. It's like a big ball of cable. Okay, look at a cancer cell. Again, you don't have to be a scientist to see that this is different. Same exact treatment, but the green signal just does not spread over the mitochondria because the mitochondria are fragmented. They're not connected because there's too much drip, not enough mitofusin. And here's a cancer cell that we've treated with an inhibitor of drip. And now you can see the mitochondrial network is reestablished. And so, as a reward for listening to this, what does that do therapeutically? Well, probably not surprisingly, here's the control group, a bunch of tumors from rats that didn't get any effective treatment. And here's the ones that got treated with a mitofusin 2 virus, and they're much smaller. Here's the control tumors that didn't get treated with anything to block drip. And here's the ones that got treated with a small molecule that blocks drip. So we haven't taken this to the bedside yet, like we did with the DCA story, but this would be the step before that. So this is what happens in lung cancer when mitochondria attack. Now, because you've been such a nice group, I'm going to show you a movie that's going to make your day. So this is, of course, just my perspective. And let me tell you that not everyone has the same perspective, and perspective matters. Let's see if this works.
Scusi, buon uomo. Mi sa dire l'ora, per favore?
What time is it?
Le 5:35.
Tells him the time.
Ma è sicuro? 36. Grazie, prego.
So he drives away, and sometime later, he returns.
Ha mangiato il ciondino, eh? No. Senta, mi sa dire che ore sono adesso? Androlta. 7:15. Oh, ma sono le 7:15 spaccate. Incredibile.
How can you be so precise?
Ma lei come fa a leggere l'ora nelle palle del ciondino? Ma io le alzo. Sotto c'è il campanile.
So it just may be my perspective that mitochondria are the center of the universe. Happy that I've taken the questions.
Thank you for coming today and for your support of the Women's Giving Circle. And please remember to consider any friends, family, or co-workers that might be interested in joining our circle. We have information available through brochures and the UHKF website. Knowledge is power, so if you can please help us get to that magic number of 100 women, that would be greatly appreciated.